Welcome to Matching Biomarkers to Disease Biology and Therapy. During this seminar, all participants will be in a listen-only mode, and there will be a question and answer session at the end of the presentation using questions submitted through the Q&A feature. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild. I'm the Executive Director of the Life Raft Group, and I'd like to introduce our presenter for today. Dr. Jason Sicklick is an internationally recognized surgical oncologist who specializes in GIST, abdominal, retroperitoneal sarcomas, and liver tumors. Dr. Sicklick is a champion of innovation and collaboration to improve outcomes for cancer patients. He's the leader of the sarcoma disease team and co-leader of the structural and function genomics program at UC San Diego's Health Moores Cancer Center. He is also a member of the NCCN Soft Tissue Sarcoma Committee and GIST Subcommittee, which develops best practices for treating these rare and often challenging to treat cancers. As professor of surgery and pharmacology at UC San Diego School of Medicine, Dr. Sicklick conducts basic translational and clinical research to improve our understanding of GIST, as well as to enhance precision medicine approaches for treating metastatic cancers. His laboratory focuses on the molecular mechanisms of GIST development and drug resistance. And I'd like to hand this over to Dr. Sicklick. Thank you for joining us.
Thank you very much, Sarah, for the invitation. It's a pleasure to be here today. Let me share my screen. Is that coming through okay?
Yes.
Okay, perfect. Okay. Well, thank you very much again for the invitation. And as Sarah mentioned, I'm going to be speaking about matching biomarkers to disease biology and therapy. So by way of a little bit of background, I just before we sort of get into the specifics related to GIST, I want to discuss what is a biomarker. And so the term biomarker, which is really short for biologic marker, refers to a broad subcategory of medical states observed from the patient, which can be measured and accurately and reproducibly. This includes everything from using biomarkers of response to a treatment, to biomarkers that can predict response to a treatment, to biomarkers of safety, that a drug may or may not be safe for an individual person, for instance, biomarkers of susceptibility to developing a disease, other diagnostic biomarkers, or ways to monitor somebody's disease and or their responses to therapy. And this can be lots of different things. And this is by no means a complete list, because there's certainly more and more evolving approaches. But what these biomarkers can include is avenues for diagnosis, prognosis, and prognostication between pathology, toxicities. For instance, there's drugs for lung cancer that actually portend that if somebody develops a rash, you actually know that they're more likely to respond to the therapy. Imaging, blood biomarkers, patient demographics, meaning age, sex, race, gender, etc. Genomics, meaning at the DNA level, or proteomics, meaning at the protein level. All of these factors can come together, whether individually or in combination, as part of determining biomarkers for an individual disease. So the first question we ask is, are there biomarkers for GIST? And so just a little bit of background. So we know that the interstitial cell of Cajal, or the pacemaker cell of the GI tract, is believed to be the source of cells that gives rise to GIST. And these are some smooth muscle type cells and fibroblastic-like cells within the gut that regulate peristalsis, or squeezing of food along the GI tract. In this way, it's an involuntary thing that we don't have to think about when we eat. And so it's believed that these cells are the ones that give rise to GIST, arising within the wall of the gut, and therefore leading to tumors that occur within the wall of the stomach or small bowel or rectum, etc. And these tumors are marked by two different markers. One is c-KIT, also called KIT, or CD117. And then the second one was originally has been called ANO1 or actomamine 1 or TMEM 16A, and was subsequently given the name DOG1 or discovered on GIST 1. And so between KIT and DOG1, those are our two principal biomarkers for the histology of GIST, meaning histology referring to the pathology of a tumor. So as a starting point, we can say that there are biomarkers for GIST, one being KIT expression and one being DOG1 expression. And these same markers are expressed by the interstitial cells of Cajal. So if we now kind of move forward in time, what was discovered in the 90s or the late 90s was that not only do GISTs express KIT, but they also have mutations in KIT. And KIT is a receptor on the surface of cells here that leads to signaling downstream through these pathways, including the RAS BRAF pathway. And so the original discovery of KIT mutations was it was a specific biomarker for GIST. And why that's important is because back in the late 90s, there was a drug that was being developed. It was initially called STI-571, and it later became called imatinib or the eventual brand name of Gleevec, that inhibits KIT mutations in cells or in vitro, and then therefore block the growth of GIST cancer cells. And so in March of 2000, the first GIST patient was treated with imatinib and had a dramatic response to therapy. And this is from the New England Journal of Medicine paper published by Heikki Joensuu, where you can see that all these black spots on the left side in panel A are tumors that are lighting up on the PET scan. And the PET scan basically uses sugar that's molecularly tagged with a nuclear tracer that can indicate whether cells are actively utilizing glucose. And what you see there is that the tumors on the left side were dramatically responded to therapy on the right side when treated with imatinib. And this was the first signal that imatinib could be used to treat GIST. And so what ensued after was this sort of drug-centric precision medicine approach. Precision medicine meaning that we're sort of have a molecular target or a molecular biomarker, and we've got a drug. We put them, a match together, and we target it. So at that point, we sort of made this presumption that KIT was a marker of GIST, and that a lot of patients had KIT mutations in their tumors. And so all these patients would receive imatinib as the drug of choice for GIST. And so what ensued over the next couple of years was pretty miraculous. This is a very rapid timeline for going from discovery of a drug, treating a disease, to FDA approval. But as you can see in the slide, from July of 2000, it went on to go to the phase 2 trials, and then phase 3, and then ultimate FDA approval for unresectable and metastatic disease in 2002, landing on the cover of Time magazine as this new magic bullet for cancer. But at that time, we kind of had this presumption that 100% of GISTs were driven by KIT mutations. And so, we start adding on a second or third biomarker of GIST, not being the KIT mutations in the DNA. And at that point, what we would say is that having the KIT mutation would predict that the patient's tumor would be sensitive to imatinib. And so what's ensued over the last 20 years is development of additional lines of therapy. All drugs, from first line imatinib to second line sunitinib to third line regorafenib to fourth line ripretinib, all drugs that target KIT, the yellow dot there. And each one is a slightly different hammer targeting KIT, if you will. And you can see there is the data from the clinical trials of the median progression-free survival, meaning the amount of time before patients progressed on treatment. And you can see there, first line progression-free survival is about 19 months, but as soon as you get to second line and beyond, you're really only talking 5 to 6 months of disease control before patients' tumors start growing again on these drugs. Additionally, you can see there the ORR, the objective response rate, basically tells us what's the response rates of how many patients had greater than 30% shrinkage of tumors on treatment. And so you can see about half of patients in the first line, and then we get to about 5 to 10% in second line and beyond. So suggesting that we are making progress in the field, but that there still is room to grow. That said, these drugs are all targeting the KIT target as a biomarker. But as time has gone on, we have begun to learn more. We've started learning why that these drugs begin to fail. And so this figure shows the KIT protein within the wall of the cell. And the proteins or genes are split up into what are called exons. And exons are little parts of a gene, sort of, if you will, like cars on a train that all come together. And so we start off at exon 1 and go on through exon X, depending upon the gene. And so the principal areas where we see mutations in KIT are exon 9, exon 11, exon 13 and 14, and exon 17. And these are often either the primary mutations, meaning that these are the original mutations that the cells develop most commonly in exon 9 and 11, or secondary mutations, meaning that often with treatment time on treatment, the patients will develop tumors that gain additional mutations that portend that the drugs will not work for them. And so what you can see here in the drug sensitivity, the green being sensitive, the yellow being intermediate, and then the red being resistant, is that each of the drugs has a different spectrum of sensitivity. And as we sort of go farther down the line there, what you can start seeing is that the exon 17 and 17 mutations cause resistance to drugs like imatinib and sunitinib, versus we see some of the resistance with regorafenib and ripretinib creeping up mostly in the exon 13 and 14 space. So it's through this information that these knowledge of these mutations and which exons they arise in actually begin to form an additional biomarker within the KIT gene itself to provide us with additional knowledge. Furthermore, we actually know that even within the KIT gene, the mutations actually portend different sensitivities to drug. And so one of the sort of great discoveries, and to my knowledge, this is the only disease where we actually will use a genetic mutation to dictate what drug dose we might recommend for a patient, is that patients with an exon 9 mutation may do better with 800 milligrams of imatinib, as opposed to patients with KIT exon 11, 400 milligrams should be sufficient. So it's an interesting discovery that actually gene mutation actually dictates potential drug dosing. So now we add on a new biomarker. We add on the biomarker that KIT mutations in the DNA can predict sensitivity and resistance as well as dosage. So where did we go from KIT was the next discovery in the early 2000s by Dr. Heinrich and his group, was that a subset of GIST were driven by PDGFR alpha, or platelet-derived growth factor receptor alpha mutations. This wasn't very surprising, just given that there's a very strong similarity between KIT and PDGFRA, and they signal through the same pathway. And in fact, actually, if you look on the right side there, this is the PDGFR alpha protein, and you can see there the different drugs and the exons regarding the sensitivity and resistance to drugs as well. And so we can start sort of seeing that similarly, that there are mutations within PDGFR alpha that are sensitive to imatinib, sunitinib, regorafenib, and the latest drug, which I'll come to, is avapritinib or AV. But there's also mutations that are resistant to these. And so now we add on an additional biomarker being the KIT, and PDGFR alpha mutations in the DNA predicting drug sensitivity and resistance. As time went on, we began to learn more. Not surprisingly, if you go one step downstream of the KIT and PDGF is the RAS and NF1 pathway. And so discovery that patients with neurofibromatosis, which is a hereditary syndrome that can cause cancers, including GIST, patients with NF1 mutations can develop GIST as well. A year later was the discovery of germline or hereditary SDH mutations in the succinate dehydrogenase genes, SDH A, B, C, and D, which are all associated with GIST. Subsequently, returning to our understanding of this signaling pathway downstream of KIT and PDGF is that just one step past the RAS NF1 step there is BRAF. And so mutations in BRAF at the V600E mutation site, which has also been strongly associated with melanoma, can also lead to GIST. And so just as an example, here, this is a patient with a BRAF V600E mutant GIST. In this patient, this was published in 2013 by the group at MD Anderson. The patient was treated, and this was the original case report of a BRAF mutant GIST that was treated with a BRAF inhibitor. At the time, it was a drug that was still investigational in clinical trials, but ultimately went on to receive FDA approval and was called dabrafenib. And so what you can see here is this modest response to treatment. But this was a drug that worked for a disease where the patients would be resistant to imatinib. And so we have now actually a couple of different BRAF inhibitors, including dabrafenib and trametinib. And what's gone on over time is actually we've learned that there's steps downstream of BRAF being MEK, which is another immediate target downstream. And actually, it turns out in combination, targeting BRAF via targeting both BRAF and immediately downstream at the MEK site with drugs like trametinib, cobimetinib, or binimetinib is an additional approach for treating these BRAF V600Es. And in fact, that's FDA approved in combination for patients with BRAF V600E mutations in any kind of tumor type. So again, as our understanding of biomarkers improves, we can start figuring out what are the best drugs for patients. Going further in time, now 6 years later, in 2014, Keith Killian and Paul Meltzer's group at the NIH discovered that there was a subset of patients with sdh-deficient GIST, which I'll get into a little bit more, that also had no mutations, no hereditary mutations, but had changes in the DNA themselves, which caused the genes to turn off. And so this brings us to the SDH patients. And so we know that, as you can see on the bottom row there, that KIT and DOG1 are two biomarkers of GIST. But we now add on a third biomarker being the SDH expression, specifically the SDHB expression. So any tumor that has a mutation in the SDH, A, B, C, or D, as well as a tumor that has this methylation or these SDHC epimutations described by Meltzer and his group that cause the genes to get turned off, if those are the underlying drivers to these tumors, then the tumors will lack expression of SDHB. So they'll be SDHB negative, as you can see here, as opposed to a sort of more standard run-of-the-mill GIST that has a KIT mutation, for instance, will be SDH positive. And so what we can start sort of breaking down is adding a third staining biomarker of GIST that we need to study. And so this SDH loss indicates that these patients have a sdh-deficient GIST. And so again, these come in two different subtypes. There's the Carney-Strattakis syndrome, which is caused by germline mutations, meaning hereditary mutations. These often occur in late teens to the 30s and are associated with paragangliomas, which is a different tumor type. The Carney's triad are these other ones, these SDH epimutant ones, if you will, that are caused by silencing of the SDHC gene by what we call promoter hypermethylation, meaning these black marks that tell the gene to be turned off. And so these are sporadic, meaning that they're not inherited. We don't know why they occur. They generally only occur in teenage girls. They also get paragangliomas, in addition to another tumor type, which is benign, meaning not cancerous, which is pulmonary chondroma, or sort of cartilaginous tumors of the lungs. And so with this, we now add on, beyond just KIT mutations, we start adding on additional gene mutations in the DNA that can predict drug sensitivity and resistance. And we add on sdh-B expression as a diagnostic biomarker. And then finally, we sort of, as time has evolved, we started understanding that there was a small subset of patients that were called quadruple wild type, meaning they were normal for KIT, PDGF, NF, BRAF, SDH. We identified that these patients have gene fusions, meaning where two genes come together and one causes the other gene to turn on. So two of these one of these is the ETV6 NTRK fusion. And it turns out that there's actually now two FDA-approved drugs for NTRK fusions. And what you can see here, this is what's called a waterfall plot. So anything that goes below that 30% line there, that dotted line, is considered a response. And so you can see that these are all patients with NTRK fusions of different types of tumors, being thyroid, colon, melanoma, et cetera, including the blue ones being GIST, where you can see that patients have dramatic responses to treatment if they have this specific biomarker. If they don't have this biomarker, the drug does not work. And it's also notable that it's important that this is a fusion, meaning joining of two genes and not just a mutation or a typo in a gene, because this drug does not work for those types of mutations. And so in this original report, they had about 3/4 of patients had responses to treatment. So that's pretty dramatic. And then the last gene is the FGFR, or fibroblast growth factor receptor genes. And again, it's another gene fusion subtype that can be targeted with FGF inhibitors. So moving on past the genes, what about other things? What about things that we might understand even at the pathologic or histological level? One of the things we know is that not all GIST look alike, even under the microscope, even before we talk about the mutations. What we know is that the KIT mutant GIST tend to be spindle, or sort of this kind of wispy looking here, or a mix of spindle with what we call epithelioid, which is what we often see with the PDGFR alpha or the SDH ones, versus like the NF1 mutant GIST tend to be more back to the spindle type look that we see with the KIT mutant GIST. And it's only, again, kind of putting this all together where we see the SDH ones that are negative for the sdh-B staining. So we now add on a sixth biomarker, being histology, that we know has some degree of correlation with gene mutations. So taking another step beyond is, what about the anatomic locations of GIST? So we know about half of GIST occur in the stomach, about a third occur in the small intestine, and then a small fraction occur in the rectum and the colon. But what we've now begun to learn is that actually the mutations that we've talked about actually correlate with different locations within the GI tract. So the stomach tends to mostly have KIT exon 11, PDGFR alpha, and SDH mutations. The small intestine tends to be a combination of KIT exon 11, as well as now we add on KIT exon 9. Plus, we see that the NF1 mutations, whether it be the germline or the sporadic ones, we can sometimes see now that we've discovered. If we look at a specific spot within the GI tract or the small intestine called the ligament of Treitz, which is where the first portion of the small intestine called the duodenum becomes the jejunum, This appears to be a hotspot for KIT mutations, as well as BRAF V600E mutations, as well as NF1 mutations. And so we can start understanding more about the different mutation types based on the location. So it's, as you can start seeing, there's not one biomarker of anything, but it's a matter of putting all these parts of the puzzle together. So are there other biomarkers for GIST? So the answer is we can start saying that location is another biomarker that has some degree of association with gene mutations. Now we can learn something from other diseases. So if we look at, this is data from gastric adenocarcinoma, or gastric cancers that arise in the lining of the stomach. And what we've learned, this is from the Cancer Genome Atlas, or TCGA is what it's called, that different parts of the stomach, for instance, actually have different causes or frequencies of different causes that lead to gastric cancers. And so, for instance, you can see there that if you look at the top part of the stomach, this tends to be this chromosome instability subtype that's about 2/3 frequency, versus it's less than half at the bottom part of the stomach. And vice versa, you can see there that the microsatellite unstable population only represents 9% at the top, but it's about 1/4 at the bottom part of the stomach. Furthermore, we've also learned that there's other types of diseases. So for many years, we thought that all bile duct tumors that arose in the liver or outside of the liver, in the bile ducts that drain outside the liver, or the gallbladder, which are all in continuity there in green, all were the same. But it turns out they're all have completely different biologies. Even more sort of familiar to a lot of us is colon cancer, which is incredibly common. It's the third most common cause of cancer in the United States. But what we see is that the genetic makeup of the mutations on the right side of the colon, here in red, is completely different than those on the left side of the colon. And what we've learned is that the biology of right-sided colon cancers is often very different than left-sided colon cancers, even though we sort of think in our minds that they're all the same. And so what we're starting to put together, just like I just showed you a moment ago for GIST, is that the biology of tumors is partially dependent upon these factors that we've discussed, but also clearly we can start thinking about linking them to location. And so we hypothesized that actually a similar effect may be occurring within the stomach, which we know is the most common part of the most common location for developing GIST. And so we developed a collaboration between our group, as well as the Netherlands Cancer Institute and Oregon Health Sciences University, to look at patients with gastric GIST or stomach GIST, and look at these patients, look at their tumors and what's causing their tumors. And so we pulled together all of the data from all these patients. We included in there this histology, or these different subtypes, the spindle, the mixed, the epithelioid. And what we discovered is actually very similar to what I just showed you for the gastric adenocarcinoma, is that there are differences in the types of tumors, or the mutations in tumors, depending upon what part of the stomach. So the top part of the stomach, which is what we call the cardia and the fundus, the cardia is right where the esophagus comes into the stomach. The fundus is this upper part here, tend to be almost completely KIT mutant in dark blue there. As we move farther down, we see only about 75% of GIST having mutation in KIT along the lesser curve or the greater curve of the stomach. But we start seeing the PDGFR alpha in orange here, and the SDHs in gray starting to crop up here. Interestingly, as we go all the way to the bottom part of the stomach, what we call the antrum, right before it becomes the first portion of the small intestine we call the duodenum, we can start seeing that only 1/4 of the tumors have a mutation in KIT, but we start seeing this dramatic increase in the number of PDGFR alpha mutations, as well as a persistence of the SDH mutations. So for instance, we quite rarely see an SDH tumor occurring up here. We essentially only see them occurring down here, and the same thing goes for the PDGFR alpha mutant tumors. And analogous to what I just showed you with regard to the histology or the different cell types, we can sort of see that the KIT mutant ones, which tend to be more mixed and spindle and yellow there, tend to be at the top, versus we start seeing more epithelioid tumors in blue as we go more towards the bottom, which is consistent with the sdh and the PDGFR alpha mutant GIS. So the histology follows the location, as do the mutations. And so this is just further analyses that we did, which basically show that we can start predicting what the mutation is, whether it be a KIT mutation or a non-KIT mutation, based upon just a combination of the location within the stomach, as well as the cell morphology under the microscope. And so we can start now being a little bit even more granular in terms of the different likelihoods of a mutation within a given area. Why is this important? I'll just give you an example. A while back, I saw a patient that was seen at an outside hospital, was diagnosed with a GIST right at the bottom part of her stomach, was recommended to undergo treatment with imatinib. I saw her in second opinion, and at that point, I looked at her histology and said, "This is a tumor at the bottom part of the stomach. It's occurring down here. It has an epithelioid histology." I said, "This is more than likely not going to be a KIT mutant GIST. This is more likely to be a PDGFR alpha mutant GIST, because the staining for the sdh was normal or positive." And so we sent her tumor off for sequencing, and it turned out, lo and behold, she had a PDGFR alpha mutation that portended that she was going to have resistance to imatinib. And so we saved that patient being treated with a drug that has the potential for causing toxicities or side effects, but has no potential for efficacy, meaning improving the response or the treatment of her tumor. And so knowing the simple fact of these correlations between mutations, locations, and the histology, which is easy to get when we've got a biopsy, can be important in decision makings as a biomarker for patients. And so now we add on an eighth biomarker for GIST, being not just GI location having an association with gene mutations, but the specific area in the stomach having an association with gene mutations, which we know correlate with drug responses. And so this brings us to a drug that we can use for PDGFR alphas. And this was a drug that was just FDA approved a couple of years ago. And it's called avapritinib. And this is a drug that you can see here in this waterfall plot. Anything, again, going below that dotted black line going downward is considered a response. And you can see here, 86% of patients had a response to therapy, and 95% of patients had some type of tumor reduction. So a pretty impressive response to treatment. If you recall back when I first presented the data earlier in the talk, for imatinib, for instance, the response rate was only about 52% in the original trial. And so we can see here this dramatic response when we start picking out specific subtypes of GIST that have specific mutations that portend that they're going to respond to a specific drug. And so what are the clinical implications of all this? So the clinical implications are that GIST are not a homogeneous disease, that they can possess different mutations, somatic meaning that mutations that occur within the tumor itself, that can correlate with tumor location as well as cell morphology. And so, in an ideal world, we would get next generation sequencing on all patients. Sometimes there are countries where that's not possible. There's sometimes when time is not of the essence and we can't wait for the genetic testing to come back, and we need to use these context clues that we have from all these other additional biomarkers. We can start teasing out, potentially which patients have a KIT mutation versus which ones don't, and therefore which ones are going to be more likely to have TKI or tyrosine kinase inhibitor, meaning drug sensitivity or resistance. But in the perfect world, we would have all patients getting sequenced. But unfortunately, that is not the case as of right now. This is data from Jonathan Trent's group at University of Miami. This is data that was published several years ago now, but what you can see there is that this was national level data looking at genetic testing in the United States. And what you can see there with time from 2010 to 2015, there was a slight increase in the number of patients getting genetic testing, but roughly only a quarter of patients in the United States are getting genetic testing of their GIST as of 2015, meaning that we can do better. And I know Life Raft has had a very strong campaign over the years to advocate for doing genetic testing in patients. We certainly advocate for it in our practice, and it's now as part of the NCCN or National Comprehensive Cancer Network guidelines that all patients with GIST should receive genetic testing when considered to be appropriate in decision making for treatment. And we took it a step further. We actually asked a question, and I'm not going to get into this in strong detail, but we actually did cost effectiveness analysis modeling to ask the question of, was mutational profiling and tailored use of imatinib cost effective compared to just giving everybody imatinib for all patients that presented with metastatic GIST? Presuming that everybody would get 400 milligrams of imatinib just as a blanket of they've got GIST, versus should we tailor their therapy based upon genetic testing, where patients with exon 11 mutations would get 400 milligrams, patients with exon 9 mutations would get 800 milligrams, or if they had neither of those, they would get no treatment. And what it turned out was that there's been lots of concerns about the cost of genetic testing being the sort of barrier to obtaining genetic testing. And it actually turned out it was actually more cost effective to do genetic testing and actually tailor therapy based upon those results than just to give everybody the same treatment irrespective of what their mutation is, up to a cost of genetic tests of about $3,700, which actually falls beyond what the standard most companies charge now, which is generally about $3,000 for genetic testing. So what this suggested is that using this modeling is that the cost of next generation sequencing and tailored therapy based upon this biomarkers that we can discover with genetic testing was cost effective, and it should be employed in our approaches to treating patients with GIST. But this is all obviously under the recognition that not everywhere in the world has as easy access as we do, and even within the United States, there's not equal access across. Unfortunately, these are countries where there's nationalized healthcare systems where often they cannot get genetic testing done, and so that's where we can sort of fall back on some of the biomarkers that we've discovered, including very simple ones like knowing where the tumor is located, in order to help us guide our management of patients. So what we've seen over the last several years is that we've been slicing the pie of GIST thinner and thinner, including using molecular testing to do it. And that in 2000, GIST really set the precedent for what we consider now to be precision oncology with the development of a molecularly targeted drug against KIT. And over the last 22 years, it's become increasingly evident that the biology and the treatment of GIST are more complex than it was originally appreciated, in that we're now learning that there are many factors beyond just the presence of KIT expression that can play a role in understanding the biology and predicting treatment responses and failures. But ultimately, when available, we would certainly, are strongly advocating for molecular testing, but we don't always have that opportunity. As I mentioned before, depending upon where somebody is in the world, one, two is sometimes we don't have the time to send off for genetic testing because of somebody having rapidly progressive disease, and we might have to use all these other context clues. Or the tumor might be in a location that's a challenging area to get enough tissue to send off for genetic testing. So sometimes in a perfect world, we would have all this information. Sometimes we don't. And so we have to use these other biomarkers as approaches in our decision making. So where is GIST going in the future in terms of biomarkers? Well, there's several different things that are under study. One of those is using circulating tumor DNA, or basically DNA shed by tumors in the blood, to find mutations instead of using biopsies. This is still an evolving field. Even what we see now, based on what's been published in the literature, is that only very large tumors and those patients with very advanced metastatic disease, only about a third of those patients tend to have DNA shed into the blood. So it's not a perfect test yet. It's helpful when it's positive, but it doesn't mean anything when it's negative in terms of the results. There's now been data published using artificial intelligence and imaging to predict recurrence after GIST based upon using the CT scans. There's other studies that are looking at other genes and expression of genes to predict prognosis. And for instance, the risk of recurrence after a tumor resection, and so that we can start being a little bit more personalized to the individual and to predict their risk of disease coming back or not. There's work discovering novel drug resistance biomarkers beyond these secondary mutations that I mentioned early on that can predict drug sensitivity versus resistance. And there's discovery of novel drug targets in addition to the classic drug targets that we've thought about, being these KIT inhibitors and now the newest PDGFR alpha inhibitors, or drugs against BRAF and NTRK. So the promise of precision medicine was to sequence patients, to stratify them, and to molecular match them. And so we've gone from these targeted therapies where everybody was just considered to be the same type of GIST based on KIT and DOG1 staining, where everybody got imatinib, to sort of smaller subgroups where we could start being a little bit more specific, saying somebody had a KIT exon 11 versus a 9, and we're going to give them 400 milligrams versus 800 milligrams. So now we're moving towards more personalized approaches where individuals can be treated on a one-off N of 1 basis based upon the DNA and the RNA in their tumors, that then can predict whether they might receive imatinib versus avapritinib for the PDGF mutation versus dabrafenib and trametinib for a BRAF V600E versus one of the two NTRK inhibitors, larotrectinib or entrectinib, for an NTRK fusion. So how do we even take this a step further? And so one of the approaches that we've done in San Diego that Sarah mentioned is sort of this personalized medicine, patient-centric approach to treating GIST as well as other advanced diseases. And so if a patient, for instance, has a mutation in KIT, but they also have mutations in other genes, can we think about developing strategies to treat these patients on a more individualized basis? And so if we look at that patient that had that BRAF V600E mutation that was treated for 24 weeks with a drug, you can see there for 24 weeks, that's a pretty small shrinkage relative to the size of that tumor. So several years later, the patient was seen in San Diego with this ongoing BRAF mutation, and we added on that second drug, trametinib, that we now had FDA approval for treating BRAF V600E mutant GIST. And we didn't see much of a response to treatment. You can see the circles around the areas lit up on the PET scan. But what it turned out, he had a mutation in another gene called CDKN2A. And there's actually drugs that are immediately downstream of CDKN2AB that can be used to target that signaling pathway within the cell. And so in this patient, we added on a drug that targets the CDK pathway. And what you can see there is a dramatic response to treatment, where these lesions in the lungs disappeared and this disease around the rectum responded dramatically to treatment. And so starting to think about not just the standard targets that we've all been accustomed to thinking about, but targeting additional alterations as well. This is another example. This is a patient that presented with a very large GIST, as you can see there on the CT scan here. I took him to the operating room. We did a resection of this tumor. It turned out to have an exon 9 mutation, and we put him on therapy. And over the next several years he did well, but developed a recurrence about 5.5 years after surgery. He was put on the ripretinib trial, which was ongoing at the time, and then failed that, and went on to be treated with sunitinib, and then had a couple of surgeries and recurrences thereafter. And we saw him at this point. We did sequencing and realized that he had the KIT exon 11 or 9 mutation, and as well as mutations in another gene called PTEN, and then the CDKN2AB similar type mutation that we just discussed with the prior patient. Well, what we know is that there's been studies of a combination of imatinib with another drug called everolimus that targets the PTEN pathway here. That's been studied in phase 1 trials by Patrick Schofsky's group in Belgium, and shown to be a safe combination. And so we took this combination that we know has been shown to be safe, put this patient on it, and you can see he had a dramatic response to treatment by targeting the additional alterations that the patient had. And so where do we start going from here? Is we need to start thinking about how do we personalize each patient's individual disease and their treatment in a more biomarker-driven approach, rather than a one-size-fits-all for KIT mutant versus PDGF versus SDH, et cetera. And so we're continuing to learn. This field is continuing to evolve. There's still a lot of unknown questions, but certainly a very dramatic increase in the amount of biomarkers that we can use now, both in the tumor as well as at the patient level, to really understand more about the disease and how to treat it. And with that, I want to thank all the members of my lab, and as well as my collaborators and the patients and their families for all their support of the work that we're doing. In addition, all of the funding from philanthropy, including funding from the NIH, as well as the Miles for Merrick Step Challenge and Life Raft Group. And with that, finish and happy to take any questions.
Thank you so much. That was a great presentation. Before we get into questions, I just want to share a note with people that one of the supporters of Dr. Sicklick's work and his research is through Miles for Merrick. There's currently, at this moment, a campaign going on in the next between December 1st through December 14th, a step challenge. So if you're interested in supporting Dr. Sicklick's research, feel free to use the QR code and the link on this page to participate. So I'd now like to go into questions. One is not really related to the topic, but it seems to be of interest since COVID is still on people's minds regarding the COVID virus and imatinib, and if there's research about imatinib lessening the risk of a COVID infection. Is it best to stop imatinib if you have the COVID virus? Sorry, a little bit tangential.
I'm not familiar with that data, and I would certainly recommend, if you develop COVID, to talk to your oncologist about what is best for the individual patient, and their care. But I'm not aware of any data particularly on that.
Okay. The next question is there any biomarkers that would suggest for a KIT tumor, the dose of less than 400 milligrams of imatinib is effective, so the potential for toxicity is reduced?
So it's a great question. So there's been sort of on and off access to checking imatinib levels, the trough level, meaning the level of imatinib in the blood immediately before the next dose of imatinib. There's never been a prospective study, meaning a study looking forwards in time to follow each patient to prove that a lower dose, is as effective. But that being said, what we know is that a trough of 1,100 milligrams per, I think it's deciliter on the units, is an effective dose. And so, we're all different. All of our sizes are different. Our metabolisms are different. It's very like these, when drugs are FDA approved, it's for the greater, larger population overall. And so, personalization of drug dosing is an important thing to consider. It's certainly something that can be done, and it's something that we're still continuing to learn about, but very reasonable to think about.
Okay, great. Some more questions are coming in. So, have you seen recurrence of GIST that arise in a completely different location? So, for example, the stomach the second time versus ileum the first time.
Meaning like, and I'm assuming this is talking about not recurrence being like spread of the original, but an independent tumor. And the answer is yes. I've seen a patient with a KIT mutation in the stomach and a BRAF V600E mutation in the small bowel in the same person. I've actually have one patient I just saw recently who has two different PDGFR alpha mutant GISTs in his stomach, but with completely different mutations.
Wow.
Probably never heard of that before. I had never seen it before. I reached out to Mike Heinrich at OHSU to see if he had ever seen it, because he's got a particular interest in PDGFR alpha. He's never seen it either. And so, why is that important? So, for somebody, for instance, this is not a metastasis. This is a small second tumor, that we need to manage as a individually independent of the other tumor. And so, dictating whether we should or should not operate on a patient, is important to understand this as a biomarker.
Does your study cover the standalone tumors outside the GI tract and what category they fall in?
I'm not sure the
Maybe, I have heard of like bone mets, right? With
so certainly GIST can there is something called extraintestinal GIST that can occur outside of the GI tract. I think it's still a very poorly understood entity, but that can occur. I don't know if we fully understand the mutational profile of those since they're so rare. And as Sarah mentioned, GIST can spread elsewhere. It can, in very advanced cases, not the usual, even standard sort of metastatic spread. It can go to bone, it can go to lungs, et cetera. But those are in the most advanced of the cases.
right. Oh, this is a good question. How accurate and available is the AI prediction of a potential recurrence with what type of mutation?
So, up till right now, there's never been correlation between the actual mutation and the risk of recurrence after, let's say, resection of a tumor. There's been studies trying to incorporate that into the risk of recurrence models that we have out there right now, and it's never been statistically significantly different than what we have, and that's thus the reason why we use things like tumor size, location, and mitotic count right now as our standards. That said, we're in a point in time where these
Great.
things are all rapidly evolving, and I would certainly say that probably 5, 10 years from now, I have no doubt that that's going to probably play a role in our
Yeah,
understanding. But we're just with the cusp of using AI as well as having further understanding of the complexities of disease to start being able to predict even better, who are the patients that are more risk for spread versus not, et cetera.
great. And at what time do you recommend NGS for GIST at the time of initial excision or at the time of disease progression or both?
So I think it's a great question. I think that a couple of things. One is if we're considering giving somebody imatinib or therapy after surgery, after resection, it's certainly a time when we would recommend doing it, because we don't want to give somebody therapy in what we call the adjuvant setting or the postoperative setting after resection when they may have a mutation that doesn't portend that they would respond to therapy. So that's certainly, one time. If somebody if somebody presents with metastatic disease, we certainly would recommend doing that in our practice. Not everybody does, and that's the reason why we did that economic study, was to try to advocate that it's not just about the money, that it actually is cost effective to do that.
Yeah.
It's a little bit debatable, depending on who you talk to, about getting sequencing at your recurrence or at progression of on drugs. We certainly often do it because I think that, in our practice, that trying to personalize the therapy and not just follow the cookbook, first line, second line, third line, fourth line, and trying to understand, using the science that we know about what resistance mutations correlate with drug sensitivity versus resistance, helps us guide therapy.
Oh, okay. So thank you so much. It looks like there's no other questions that came in. So I'm going to give people some time back at the end of the hour to enjoy the rest of your day. I just want to say thank you so much for the time and the presentation. I hope everybody learned a lot. And just note that this is recorded and it is going to be archived, and we're going to be posting it shortly for others to rewatch, or for those who couldn't make it to see it later. So thank you so much, Dr. Sicklick, for the presentation, and everybody enjoy the rest of your day.
Thank you so
Bye.
much for the invitation. Have a great day, everybody.
Thanks. Bye.